life on Earth, some plausible theories about how some of the different components may have arisen over time. We've entertained and realized that it's, you know, it's thoughts, it's some evidence simulated, and then a lot of theories. So, of course, that's in science a great area to maybe rethink things, and I'm sure people are continually. So we present a story here, maybe not the story. One thing I did want to go over again, just briefly, because I think I did a little bit fast, was the beginning of the first idea of metabolism, and this idea of getting some proton motive force, so ATP, through what? What kind of phosphorylation? Would it be oxidative if we're using the membrane, or substrate level?
Substrate.
Or? Oxidative, okay, think about the movement of the electrons and that we're getting phosphorus with the use of ATP in a membrane. So we had these early compounds that existed on Earth, and the first thing in this, the chemistry is a little bit not as obvious as usual, but we do get this movement of the sulfur into iron disulfide, I suppose it would be, and the generation of hydrogen gas molecules. So that part was pretty straightforward as an inorganic type of reaction. And then what we get from this then is really a very weak, kind of electron movement through the membrane. Nothing like the electron transport system that was to evolve throughout time and complexity. But it is the beginnings of things, so I guess in some ways we expect things to be simple. And so the electrons moving through, elemental sulfur exists, or existed and exists in the ocean. And so that was the electron acceptor. I don't know that I really pointed that out sufficiently the other day, and I wanted to do that. And we can tell by several different ways. One of the easiest ways is to see that we're able then to grab some hydrogen and reduce the sulfur using those electrons. So that is the, continue to look for pattern. That's what I think is important in biology. There's so much information, looking for pattern. So, this idea, well, there had to be some kind of reductase or some kind of reductase type of entity. We talked about the problem of enzymes at that time, but this is a theory, okay? We have a cytoplasmic membrane, the blue, what is it called, the blue shaded area. Okay, and then this hydrogen sulfide, at least in the cell, could be regenerated and used here. Not known yet, at least as far as I am aware, that there was a cell wall to keep this all nice and contained. So this could be a step that actually was regeneration, or it could also just be that it was expelled. Because remember, hydrogen sulfide, rotten egg smell, is very prevalent in reducing conditions. Walk through any swamp or something, and you will smell the smell. It is, to most of us, not terribly pleasant, but certainly a little bit lets us know it's there. Okay. Then we have kind of the rest of the story. I mean, the rest of the story assumes that we have another type of entity, and that is then the primitive ATP. I already have it. Primitive ATPase. So again, another enzyme that, how did that come about? Well, billions of years, not much to do, I guess. Things happen, although not terribly clear. But then here we have, once those electrons are removed, we have hydrogen ions then can move through. And this is very familiar to us from our earlier study then. We see that, we get some kind of proton motive force by this very slight buildup of the chemoosmotic effect of hydrogen ions to the external portion of the cell membrane, our kind of layer in this biological battery that we've gone over several weeks ago. And this can move through then, bring hydrogen in, the hydroxyls that were generated over here can then be reduced. We generate water, but we get ATP, okay, the phosphorus currency. Could that exist? Well, if there's nucleotides, yes, it can, and so this is the idea, at least, of one plausible explanation using sulfur-based metabolism to cause a generation of an energy source as we know it now. So, billions of years ago, did this happen? Well, this is the idea. How so? This is kind of, in my mind at least, when we take apart cells and species that we found at the base of the tree of life, they see some of these systems and you can project one way or the other from that. But if you go back and say, well, Thermatoga and Thermus aquaticus have some of these kinds of things that look like this, maybe they evolved this way. I think that's kind of it. When you pick up a fossil and you look at a trilobite, you go like, huh, kind of looks like something that exists today, but what is this related to? How did it come about? It's evidence somewhere from the past. We know it's in a rock, right? So this, not necessarily in a rock, but maybe the living genes, the living organism has that inheritance story from the past that we can look at. So maybe obvious to a lot of us, but I don't think it hurts to restate some of these ideas as we think about how do we know what we know. Okay, and then we got to this idea, with energy that's being able to be produced, with the idea of a cell, with moving towards DNA as the central inheritance and kind of, in a way, a memory molecule, if we kind of think of it in computer terms. We have the stage about at this time to start launching the domains. Now, this in itself is not a straightforward idea. They just didn't suddenly like, boom, do this, but it's speculative. So, we move to DNA-based life. Organisms with small genomes. We have evidence for that as we go back and remake the phylogenetic tree based on inheritance. Okay, we can note that going back to things like Aquifex is another genus at the base. I haven't really talked about it too much yet. And the domain bacteria. We can see that from our early organisms that are hyperthermophilic. Okay, that kind of fits our scenario type of story. And then, still this question, well, there is no nucleus. Where does the nucleus come from? And this is something where if you go into literature, there's some nice evidence about the nucleus, and we'll talk about that. We already talked about this. As just a memory jog, we see that some parts of the membrane, which is an imperative part towards life, and we talked about how important it is to have what? Compartmentalization, to have, even on the level of a cell, we know the eukaryotic cell became much more compartmentalized, old news to everyone here, but that really localized and allowed things to happen, but that cell membrane, could separate inside from outside, whether it started as a micelle or something else that was a really big thing. And then to put into it, to interject that kind of biological battery, that idea that you can make the outside more positive, the inside negative, and move things through, is a big deal. But, for different reasons, and I'm guessing the thermodynamics of it, and the inheritance of it, that is, where were these things evolving? What was better adapted for living in hot, smelly, anaerobic places? Probably two types of lipid patterns started to show up. Okay, the Earth wasn't all the same as it is not all the same now, but probably, I would say, in some ways less diverse. And so you probably have selection for two different types. Okay, oftentimes too we see neutral selection. If it really doesn't change anything, it just it is adopted and moves on. There's kind of this neo-Darwinian concept of neutral selection. But I am guessing, because of conditions that we've talked about, stability that is given through the isoprene units by having a phospho type of lipid that isn't necessarily a bilayer. A lot of those different things probably afforded different adaptations, and maybe just started out, on that level, being selected for survival. Those selected for it, and later were carried on. We also do note in subsequent times from whenever this early adaptation happens, that now, and we'll get through it either in this lecture or next, that the lipids of the lipid or the cell membrane are quite variable even now. Not everything has a phosphoethanol amine, like we showed for the E. coli. There's serine groups, there's all kinds of things in there, so it's something where there continues to be variants, much like we see the variants in the DNA, and actually can be used to help type bacteria. We'll get into that a little bit in a little bit later. This is just talking about the Archaea kingdoms. They are largely under investigation. Two kingdoms are pretty well established, probably started out early on and have made it through the eons. The Kryn Archaeota, you heard Los talk about that, or I think it was in the video. The Euarchaeota, the true Archaeota. Names that really right now don't mean a lot to us, except that we see things like methane in these names. We see things that are hot and full of sulfur, and those are in there. And as you'll learn more now, that's kind of how we started to typify this domain. We now realize that there's archaea in many places, flowing down the Sangamon River, and in the soil, and some are even in use. So it is an investigation. And I don't know if I brought to this class yet, but I'll try to bring to you the evidence from last year from some of the deep sea work, where it looks like they may have found questionably two more domains that seem to be related to the Archaea. So think about not a 3-domain system, but possibly a 5. Well, it's not hitting your textbooks yet. It'll take several years for that, but people are thinking about that. The small things in the ocean have gone overlooked because they're very small and the ocean's very, very big. But people are starting to find new things. I throw that out as maybe interesting to people. Thermus aquaticus, we've talked about that. In relation to what? Where did that come up? This bacteria, what's that? PCR, yeah, TAC, you can see very easily the T and the AQ. TAC polymerase, this thing living in a hot springs. We don't have to worry about denaturing under the conditions of PCR. You can probably denature or destroy every protein, but this was something that people figured out. But we also started to look at the beginning of the diversity of what they used to call the U bacteria, now just called domain bacteria. You may see it called that. It is somewhat confusing, but it's starting to settle down for a few years. U bacteria, Archaebacteria, it was driving people a little nuts. People are arguing that out more and more. So these are just like the main phyla that we see here. One thing to draw from this, they're a little better explained in the Archaea. Okay, maybe in 10 years, maybe whenever, another generation, your kids are taking a class, maybe the Archaea tree will look a lot like this. I don't know, probably won't be around to see that. But the diversity here we find, comes out of, our interest in disease, survival, and then that we can culture these things a little bit more. We hadn't really thought about culturing things in boiling water and stuff like that. I mean, we as scientists, not we as like my multiple personality drink. We know a little bit more about this group, and after we're done talking about evolution, we're going to start kind of making our tour through this. Okay, for some people, it's going to seem like a cool tour. Like driving down historical Highway 66 and looking at cool stuff. And to others, we'll find the tour to be kind of tedious because there's a lot of stuff, but we'll try to make the best of it. I don't know any other way to help you see the diversity and some of the cool stuff in there. So we talked a little bit about TAC, that's in there for you. So probably after the division of the domains, questionable as it was and when it was, but early on in life started to become life, as a singular cell, sulfur-based type of situation. We probably have then the divergence of the two main cell types. Okay, the gram-positive, which we now are all very familiar with, either from lab or lecture, and then the gram-positive. This diagram actually, I mean, years ago when I was actually studying how to extract entities from the extracellular matrix, but still worked meets the purpose for that. So I think the next big thing that I do think is big is, is taking energy from light. Okay, seems simple to us now, but taking energy through proton motive force through something is questionably heterotrophic, or at least chemolithotropic for sure. And it still begs the question, what are you using for carbon? Not well thought out in that early slide, but now with the use of light, we start to see the use of autotrophic life, or the evolution of that, and that by now when you see that auto idea in a length of trophism, you should be thinking CO2. I think you've been through that, you got that. So, but it's a little bit different again. It's not exactly how it is now, but we can use what all of us have been taught about photosynthesis to help us make sense of this fairly readily, okay? And of the things I had in the 10 minutes of spending time with Lynn Margulis, we talked about this probably more than anything else. I don't know why, I think we were just standing around talking. But, sulfur-based acquisition, sulfur is being used kind of instead of carbon in this case for metabolism. So, but don't misunderstand that carbon, and it's carbon dioxide, is being used as the building blocks. As we talked about, the chunk of wood, that mass comes from the result of photosynthesis. It doesn't come from, a very little part of it comes from the minerals from the soil. So what we have is an-oxygen photosynthesis, and to explain that, this just means you are not generating oxygen. Very simple, and once we look at it, you go, oh yeah, I get it. So, filamentous and oxyphototrophic bacteria are probably the first to do it. Evidence with stromatolites, which we'll talk about in a minute, from Australia. Several types of stromatolites are basically fossilized and actually existing living entities that we can learn from. So, what do we have? We have light plus hydrogen sulfide plus CO2. Now, right away, what does this look like? A lot area. Well, it kind of looks like the formula for photosynthesis if we put in water. So oh, I see, because water yields the oxygen from photosynthesis. We're going to get elemental sulfur and some kind of carbohydrate. Yeah, that's a cool thing. Okay, and so and I have that little note in there for you. So and where would you have to go to Australia now to see this? Usually if a professor asks, you go, no, but then it's he or she says, well then where would you have to go? I mean, this is common. Pick up the Winograd-Ski columns. This is going on. This is one of the things that go on. The pigments are quite different. Okay, and when I talk about that early Earth could have been purple, and probably was, or maybe more orangish, different pigments were harvesting different energy and reflecting different light, reflecting purple light and taking in more of that high energy reds and oranges, and then shifting and so forth. So pigment is necessary. You have to be able to harvest the energy to do that, and we'll kind of return to that. So anaerobic, non-photosynthetic bacteria probably gave rise to these life forms, non-photosynthetic in the sense of terrible picture. I've never really been able to find a better one. These kind of things, they kind of look like dough balls or something, I don't know, but they're the picture I could find for these anoxygenic stromatolites. What stromatolites are stone with stroma, with layers, okay? Whether we look at the modern ones or these evidence of these fossilized anoxic ones, what essentially happens is that you have shallow, in this case, ocean. And the ocean, you know, the stromatolites are down here, and the ocean has a buffer of water, or the stromatolites have a buffer of water over them. So harmful rays are somewhat buffered, you know, it's still kind of hard living, okay? But then you'll have a surface then, really, I'm not talking too much about organisms that have a way of regulating buoyancy and moving around. They're on a surface of sand or rock or sediment of some sort. They're able to get light and do their anoxic photosynthetic type of situation. But what also happens is they start to get covered. You know, the ocean's rolling around and silt comes up. And you gotta get this layer, a little bit of a layer of rock. Yeah, but you know, some of them kind of poke through and grow on that, and then they get covered. And so you make this stroma, these different layers that build up, and thus you're getting this kind of fossilized column of microorganisms, you know, along with still an active or reactive surface if they're still working. We'll look at some other ones in a minute. So, 3 billion years ago, moving what they think to the next big revolution in biology. Yay, let's go, charge. Oxygenic photosynthesis. Okay, a story we all know about here. And this story belongs to the cyanobacteria. Even though this story of oxygen photosynthesis lives now amongst our plants and so forth, and eukaryotic algae, cyanobacteria is where this idea of energy started out. We do see oxygenic stromatolites still exist today. Great evidence there. The big deal that happened was probably the first great, what would you call it, genocide? What do they call it when there's like a huge species reduction? Great extinctions? Yeah, that's a good one. There's a few other words. You get the idea. So, why? Well, we kind of know this. You know, on some level, even today, oxygen really messes up organisms that don't have a way of dealing with it. Okay, oxygen is a great terminal electron acceptor. It's a great gas, but it's poison to anaerobic metabolizers, especially if they're doing respiration of other gases and materials. Respiration in the sense of the cytochrome system, not respiring oxygen or anything. So oxygen probably caused the first great extinction. Now, that said, oxygen wasn't everywhere. It wasn't like one day, oh, we're at 21%, or is that what we're at now, 21% or so? Why did we stop at that level? Another question that I'm sure you must have asked yourself at some point. It's not clear. Why don't we have 25% oxygen in the atmosphere? Some people think it's regulated by the Gaia hypothesis we talked about. Others say other things. Anyway, so oxygen wasn't everywhere, so there was extinction, but it probably wasn't what universal pandemic, however you want to look at it. And it wasn't happening, there wasn't all that much right away. However, it was significant, and it was an idea that caught on and did very well for itself. So we get what we recognize as photosynthesis. Most of us just call it photosynthesis. Now you could be a little bit more specific and say, oh, it's oxygenic photosynthesis. And probably most people would look at, most of your biology friends would look at you unless you say, oh, you've been talking to Lemke again. You know, I see the microbes, you know, they got with and without oxygen. Okay, so that would be something to think about. Now, where do we, this is kind of cool because we do see evidence. We as people, as societies and nations, stuff, we can start to look in the geological record and see some evidence of this, and start pinpointing that 2 to 3 billion year range, okay. About 2.7 billion years ago, the cyanobacteria developed the photosystem, okay, kind of just been through that. By about 2.4 billion years ago, oxygen concentration raised to 1 part per million, 1 ppm. What is that? That's 1 microgram in a liter, yeah? Is that right? Yeah, that's 1,000, I mean, yeah, no. So, yeah, you know, about 1 microgram per liter. Well, maybe it's microliter if it's gas. Anyway, you get the idea. Part per million. I'd have to think about that a little bit. But the great oxidation event. Okay, oxygen could not accumulate until it reacted, and this is a cool one. Reacted with abundant reduced materials in the oceans. There's a lot of the iron sulfide. We saw that in earlier slides. This pyrite. Basically, a lot of iron around. And so, basically, what had to happen is that the Earth had to rust. And as Neil Young says, rust never sleeps. So anyway, the Earth had to rust itself out before oxygen could even start to accumulate. So even though oxygen is being put out, you know, you have to get rid of this stuff because it wants to oxidize. We all know that. Throw a nail in a stream and come back in a few days. It's rusty. It just wants to, will do that. Okay. And we see banded iron formations in sedimentary rocks. So I mean, this can be seen, it can be measured. It does seem to fit, because those bands are underneath other bands, and as you know, geologists and archaeologists are really good at figuring out dates and times and that kind of thing. So this gets to be a little more plausible. Hey, what is all this rusty iron doing here? Oh, well, and they kind of come up with this idea. So that fits together pretty good. Here are the little critters that did this whole thing. You go, oh my gosh, they're kind of complex. Probably didn't start out all that complex, but our main thing is that they're able really eventually to do buoyancy. Of course, they have the DNA when we think that happened earlier, and their plasma membrane really acts as a thalcaloid. The thing that you see in chloroplasts and talk about is little stacks of coins or something, whatever the analogy is there. And other things have been added probably over time. One thing that I don't know if it evolved then, but I'm guessing it might have. And I've never tracked down the story. And if you do, great. We're going to have a little discussion. You can hunt down any of this stuff. You might want to make a note. I want to hunt this stuff. But does anyone know what many of the cyanobacteria do as far as the element nitrogen? Anybody? Same kind of thing that half of the plants in central Illinois, the soybeans.
Does it fix it?
Yeah, they fix nitrogen. What does he mean by that? Is nitrogen broke? Who broke nitrogen? Did you break nitrogen, Tommy? No, he knows. Okay, so what's this fixing nitrogen stuff? Well, let's talk about that. Why sit around and contemplate when we can just move on? Nitrogen fixation, and we'll talk about this more, but is a really cool thing. Nitrogen exists in different oxidation stages of sulfur, okay? And it's kind of a paradox of nitrogen. The more reactive nitrogen is, the less of that type of nitrogen is around. So things like nitrate, ammonia, is plentiful. We can find nitrate in ammonia. We can make it. It exists in nature. But the bigger compartments of nitrogen are things like nitrogen gas. Okay, so the more kind of reactive and ones that move around with solubility and through the biochemical system, are a little bit lesser in their prevalence. Thus, they become not trace elements, but essential nutrients. So many of you may have learned in ecology or in other classes that, or many of you may be gardeners or farmers. The magic ratio of about 10 to 1 nitrogen to phosphorus is no mistake. That's what plants and us generally need to support the composition of cells. In water, that's called the Reynolds ratio. maybe talk about that. But anyway, so, we are surrounded by nitrogen. What percent of the atmosphere is N2 gas? Now, someone here should know that. You've been on this earth for a number of years.
75%?
Huh?
It's like 75%?
Well, yeah, about 75, usually about 78%, but yeah, that's what people say. Okay, so, you know, the problem is, most things can't just take nitrogen on there, even though our bodies, our cells need this. It's not a secret, we know that, you guys know. So, what nitrogen fixation is the ability to pull nitrogen gas out of the air, or wherever it exists, okay, and to reduce it. Okay, usually to an amino acid, usually to glutamine as an intermediate, but it depends upon who's doing it and how they do it. But, so you bring it down into an amine group is the point of it. Okay, these guys can also do this. I have never thought, actually until this moment, I've always just taught it, but I haven't thought that when did that process evolve? I would guess it could have evolved easily at that point, giving these guys a little bit of an edge in growing because they can get nutrients right from gas, and they have the energy, you know, from sun that can actually do it. You have information.
These the little guys that have like a relationship with plants that live in the naturals that convert nitrogen for them? That like live in the roots.
Very much like that. The rhizobium and bradyrhizobium are genera that they're non-phototrophic. They're in a different part of the tree.
I just didn't know if that was what you were talking about.
But they do the same nitrogen fixation deal in the nodules of things like the legumes, the alfalfa, soybean, those kinds of things. And even with these cells, they have to develop a specialization so that they limit, now they have to limit oxygen to allow the nitrogen reductase to work. So anyway, I suppose somewhat of a tangent, but hopefully somewhat interesting. Something certainly that continues to change the world is oxygen and nitrogen. Here are the stromatolites. You know, I think you can still go see these. I don't know if they're partitioned off from humans or not. I've never had the chance to get down to that part of the world, but you can get the idea if we took these kind of mushroom rocks, what we're looking at, and we're not talking like, these aren't microscopic, I mean, these are like you go to a cave and you see the stomatotites or whatever they are, or stromatolites, whatever. What are the things in caves, the ones that hang down and stalactites, that's it, stalagmites and tights, yeah, you know, those we know are in water and dripping minerals around and so forth. Well, these are, you know, if you cut them open, there'd be layers upon layers. That's the kind of idea here, but you got these different microbes and pigments and stuff that are captured in this. So, you know, they say, well, let's look at the stromatolites. They'll give us some evidence of how things go. We can age them, we can kind of figure out what's going on. And, oh, these guys have, you know, oxygenated photosynthesis going on, some of these other things don't. Okay, let's piece together the puzzle and make our stories and see how it works out. So, yeah, 2.7 million years ago, I think this is pretty much what we talked about. So I'm back to the oxygen revolution. Yeah, it was about the time we talked about. We had the banded iron formations, and we had this extinction to cope with. How did that affect evolution? I don't know if it's known. I mean, we were talking about, you know, destroying early microbial life. Okay, how much of it made it and found refugia, you know, to live in? I would say a significant amount. I mean, there's areas of the earth that, you know, obviously kept Thermatoga going, and some of these other things that, you know, so it worked out. So the extent of the extinction, I don't know, is well described as some of the other 3 or 4 that have gone on. So we're going to finish up our little show on evolution. Doing okay? Ready to go? All right, some people say yeah, some people are avoiding nighttime. I'd probably be asleep. Okay, so you're doing well, thank you. Okay, well, let's keep going on this. So here's kind of where we're at, looking at the picture. You know, we're not quite to endosymbiosis. That is a game changer, okay? Compartmentalization, energy. You know, somewhere in there a nucleus is showing up. Wow, that's a big deal. How'd that happen? Okay, you know, so we're sitting in this area, but what's going on? We're getting now, oh yeah, and then a whole bunch of oxygen has gone down to the bottom of the ocean. It's part of rust, or whatever you want to call it, some form of rust, an iron sulfide soup, whatever. And so eventually the cyanobacteria go, well, you know, things are rusting. I don't care. I can sit here and I got CO2 around. You know, that was in the early gas of the earth. I got some light. Things are pretty good. Don't really have ozone. Gotta be careful about how close I get to the surface, so they're gonna fry me. And I imagine a whole bunch of them did. And there probably was a fair amount of spontaneous mutations going on. But in this period then, we now, once we can accumulate oxygen, now we have the possibility of oxygen becoming a terminal electron acceptor. Doesn't it mean that before that nitrates and sulfates couldn't be? Just questionable about their abundance. Okay, yeah, it's a good idea, but you know, how did that all work out? Was the Earth too reducing and so forth? A little bit of controversy in that area. So aerobic bacteria, though, can now evolve. Very simple. No cells yet, questionably of organelles, but organelles seem to be endosymbiotic anyway. And then DNA likely partitioned into the nucleus. Now this nucleus, hang on, okay, it's coming several slides. I'll get to the nucleus then when we get there. So where are we? We are here. All right. So mineral oxidizers. Now, if we were to look for them, okay, we would look in this group called the proteobacteria. For us, maybe in this class, an idea, a group that we've talked about way early, but we know it's coming. We know it's very big, it's very diverse, and it's in the bacterial domain. That's okay for now, but that's a name that you want to start putting in your memory, proteobacteria, which has many groups. And we're looking into a group, don't worry about the number, the sulfur and iron oxidizing bacteria. And we talked a little bit about, did I talk about here? You're walking over to the HSB and you see Gallinella, the iron oxidizers right there. So we've kind of been down that road a little bit. So chemolithotrope for sulfur oxidizers. Energy is coming from some kind of reduced sulfur. You know what, you say, that one's not too reduced. Yeah, but you know how to look at the electron tower now. You know how to look at those oxidation reduction reactions. If I'm oxidizing, what is this, thiosulfate? Is that right? Did I have it right? Chemist, thiosulfate? Isn't that what this is? Actually, you're a chemist?
Yes.
Okay, let's say yes until someone says no. But anyway, it's still more reduced than this. Are you going to get a lot of energy from it? No. But that's okay, you get the idea. We got to have a place where electrons can come from. So we take a very simple example. Before we're looking at proton motor force moving through to sulfur. We had that example as we started the lecture. Here now, oxygen's a player, and so we can oxidize our electron donor and reduce the oxygen and liberate and make some proton motor force there. So in doing this, light doesn't necessarily have to be part of the question anymore, because the meaning here is that oxygen, is allowing energy to form. Okay, your other classes are great, but they're not going to be talking a lot about chemolithotropes if you're in organismal botany. It's not really a big option. Chances are you're not talking about too much. We're talking about the evolution of mammals and lizards and so forth. So this is a cool story that exists in these little single cellular now domain bacteria. Okay, so I kind of talked through this. Don't worry, if you are thinking about iron oxidizers, I'd like you to concentrate on this. This formula is pretty instinctive, I think, to all of us. iron does work also, is all you have to think in your notes. Oh, okay, iron will work, yes, it's true. Iron is kind of confusing, at least at least for me, maybe not for you, but, the ferrous thing, I always have to sit down and really think about electrons. Where here, I can just see where things are moving really easily, so I'm happy if you're with me in that camp. All right, now that, so what's going on? We get TEA of oxygen, likely, evidence. Okay. We get ozone, okay. Atmospheric kind of reaction leads to that, but okay. And so now we have a more buffered atmosphere. That's important for what we know about DNA and mutation and so forth. Things can maybe stabilize a little more. We have oxygen on Earth. Earth is cooling a little bit, actually quite a bit at this point. So we have a lot of things going on, and so we have this idea of endosymbiosis. Probably everyone here knows it. We'll not go over unless someone really is wondering what is he talking about. The ozone. Okay, so probably at this point, this is where we see probably first the mitochondria. May have taken a little bit longer for the chloroplast to go through endosymbiosis. I believe my theory slides are after this next slide, if I remember right. So where are we at? Endosymbiosis, we know it's supported. We know what it means. It's in here. Just to throw out there, endosymbiosis, probably in time, it was the free-living, probably the aerobic bacteria, likely in the proteobacteria again. That's where we seem to see it settling out. If you look at the evidence in mitochondria, where that settles out into the tree of life. Okay. A point of theory, less established, is maybe flagellated prokaryotes gave rise to the flagellated type of eukaryotic flagella. We looked at that 9+2 type of cross-sectional microtubules. You're like, well, does that really work out or not? I don't know if the evidence is good there, but we certainly have this one in the pocket, and also on the timeline a little bit later. Cyanobacteria really turn out to be prochlorophytes, which really essentially are cyanobacteria, just a little bit more of an ancestor to a modern cyanobacteria, and we get something that might look like a euglena or anything else, and something that has this crazy overlap of things. Here are protists. So, some theories on what's going on. I believe the thing to watch in this diagram is this arrow, but the idea here is when we think about what are we looking at? The tree of life, the three domains. Bacteria probably went away from this common ancestor with this archaea lineage, and the eukarya could have likely kind of come out of that. Okay, seems to make sense. The closer we get to now, more things tend to make a little bit more sense, more evident. So we have the ancestral mitochondria coming in when the nucleus has already been formed, and then later followed by the chloroplast. The existence of the nucleus is somewhat under scrutiny. Okay, the idea here, I don't know in the realm of things how much this makes a difference, but the idea here that the mitochondria gave rise to a eukaryotic cell now with more energy, more capability. Maybe that's how a nucleus was engineered in this idea of biology and evolution. Seems to be what where we're at and thinking about. I will tell you, and this is fun to look up, some people think that the nucleus may have come from the planktomycetes. If you want to write that down, it's like PLANCTOMYCES. The planktomycetes is a group we'll talk about. Virtually unrecognized like 5 years ago. Okay, we kind of skimmed over it when we talked about it in class. Now they find out that the planktomycetes seem to have little compartments in them, almost like organelles. Not quite. Well, wait a minute. Prokaryotic cells shouldn't have organelles. What are you talking about here? Yeah, life is weird. okay? Weird in the sense that it's not always predictable, okay? Could be that something about the plankton mycetes could have given rise to kind of a proto-nucleus and make something that made that nucleoid a little bit more tenable as a nuclear entity is one idea. There's theories out there that viruses gave rise to a nucleus. There's probably a bunch of other ones. As I keep hinting to, we want to do a discussion tracing down some of these things. These might be something, oh, that's kind of interesting. I'd like to look that one up. These are just ideas I'm throwing out as we talk. Yeah. So, hypotheses, well, there's it's chimeric. A chimera is something that doesn't really exist. What was a chimera in mythology? You had a head of a lion and feet of a goat, or I don't know. It had a whole bunch of body parts that doesn't exist. We still talk about chimera as non-existing things that we tend to fixate on. This is supported by several features. We do have lipids that seem to be shared. The ester linkage seen in the eukaryote, and the prokaryotes. Oh, that's kind of interesting. There's kind of that thing going on. Eukaryotes have transcription translation machinery, though they're more similar to the archaea. Yeah, archaea have histones. You have histones. Some of the transcriptional factors where proteins are started by the Tata box or the Pribno box, is shared between archaea and the eukaryote. So thus we see evidence for the branch of the eukarya off of the archaea, still as being not branched off of the bacteria, is still how it kind of adds up when you start looking at similarities in differences. I want to introduce you to Ford Doolittle. Who's an onion? I don't know if Ford is still around. He was pretty old when I saw him, but he's an interesting guy. You're in a conference, he just gets up and walks around. Maybe he has the problem that I do with sugar. If I sit down, I fall asleep. I don't know. But he is a person that speaks his mind and thinks different thoughts. Now, here is a domain of the tree of life. If you had to look at this, maybe at first you go, I have a lot of arrows there. But if you started looking at it, this should make sense to you. What do we see? We see the three domains. We see, there's some phyla, or whatever you call them now, subdomains, whatever these guys are calling them, but we kind of saw these before, and I kind of recognize these names, and here are some things we've talked about, and I have not doctored this up in the least. This is just a general diagram Doolittle puts out, and it says, in time, mitochondria probably first, probably this kind of chloroplast later. We go, that's a nice summary, Ford, thank you for that. And then he'll challenge us by thinking, just as Margulis said, just as if you had endosymbiosis going on once, what's stopping you from having a whole bunch of different interrelationships going on? In which case, students, I consider my students, the guys go like, oh no, don't do that, Mr. Doolittle, because the world as we know it goes crazy. We like some kind of order so we can tell stories. These are the scientific creation myth. Every society has a creation myth at different levels. This is our science, how life came about. It's nice to have something make sense, whether you buy it or not. At least it's good to have the storyteller tell a good story. We'll see how the story holds up. Summary, I don't think I need to go through this, but in your notes, we kind of see then these big events going on through there. Look over that. It's just a pictorial diagram of all the words that I blather out here, but you should kind of see. This is a big deal because what are the implications of endosymbiosis? Why is that such a big deal for diversity? Okay, what we call, and I want to say this term now, adaptive radiation. I used to write that on the text. Explain adaptive radiation in endosymbiosis, and people said, well, radiation will kill organisms. Okay, we got it, and certainly, yes, it will. But first off, question. Why is endosymbiosis such a big deal? What makes it a big deal in diversity, in this explosion of the tree of life? Is it a big deal? No, okay, Dr. Lemke, it's not a big deal. I could care less. Okay, well, that essay was short. Okay. But humor me. I say it's a big deal. Why might I say that?
I don't really know how that, other than like certain animals wouldn't be able to eat plant materials or things like that without end up symbiosis with different microorganisms, so you wouldn't have as much diversity between food chains and different main
Yeah,
species.
and you're way down the line. The whole idea of multicellularity, of systems, of eating and chewing, all those things are not possible at this level. Even if you took all these cells and pasted them together, you don't have that differentiation, okay? So I'm kind of putting words in your mouth, but I'm trying to bring it back to the event. So if I'm more or less fair, we'll see what other people think, but that's a good idea, yeah.
Like, with mitochondria and chloroplast, it like really changes how they metabolize.
This is the deal. Energy, think about it. Compartmentalization, key, okay? Put those little energy centers in some cells, maybe both in other cells. What does that do? Plants respire at night, photosynthesis during the day. Wow, what an incredible energy factory. What possibilities, okay? Energy in the form of light, still more or less not capped at all. We still can put up solar panels and not knock out the rainforest. Lots of light around, okay, so that's good. But yeah, this whole idea of the thermodynamics and energy just is blowing off the top. Now there's energy to not only keep yourself alive, but make different adaptations, an organism. There's energy for different adaptations. A whole bunch of them just get nailed, they don't make it. That we know natural selection is not friendly to a lot of things, okay? Even car makers. But the thing is that the energy is there for the potential, for systems, for all this stuff. So think about that, okay? And look at yourself. I mean, just look at the diversity you have, and differentiation. Not really going to work. We'll see some systems in bacteria, and they'll be pretty interesting things. They do have some clever little adaptations that have been selected for, but nothing like what can happen at this point. So when go ahead.
I just wanted to clarify, adaptive radiation is when there's...
I was just going to ask that. What is adaptive radiation?
When there's like a big boom in diversity of different species.
Yeah, radiation, this idea is adaptation and then kind of a radiation in the idea of explosion, of radiating out, as opposed to UV radiation that zaps, DNA or something. So, it's a good term. You just have to, have to look at the context of how we say it. Yeah.
Okay, so we have time to get into the mechanics of like how in symbiosis like altered the genome of the host cell. Like you engulf another form of life, but now our genomes encode for the mitochondria. So how did that get?
So the mitochondria, they reproduce with cellular division. How it's incorporated in the genome?
Yeah, how does it interact with our cells to keep?
I think what she's asking is,
Yeah.
we've had endosymbiosis and we've had the mitochondria or the chloroplast come into our cell, but how did our genome pick up and replicate so that we could then cell divide with chloroplast and mitochondria continuing in that lineage?
Yeah, how come we didn't just lose it? It interacts really well with our cells.
That's a good question. I gotta think about that. Yeah. Does anyone have an answer? It's a good question. I haven't thought about that.
Doesn't it divide? Like, doesn't the mitochondria itself also divide when the cell divides?
That's what I was thinking, but I think what they're saying is it under genomic control, is what I think might be answered. I think you're right, and that's what I started to say, but I haven't thought about the mechanisms that may or may not control it, if that's really under the cell, or if it's independent to the mitochondria.
I thought it had its own DNA.
It
Yeah,
does.
it does.
It does, but.
It's possible
But
it-
why doesn't it just divide spontaneously or on its own rule? Is there a signal coming from the parent DNA? Is that what we're talking about? Yeah.
Right.
I agree with you guys. This is about where I stop in my understanding of it. Let me find out. Or, yeah, really good question, you know? I'm not afraid to say I don't know it all. Most of you have figured that out by now. But I do enjoy finding out. So let me look into it, okay? So, rain check on that. Tables, we look at them, you know, oh, so much information. But here's what tables are for. They're to organize our thoughts. And you should look at this table and see most of the things I'm talking about. I'm using it as a summary. Have we been through the membrane lipid thing? Oh, yes, many times. But now look, the three domains are the columns. Who has it, who doesn't? So when we're talking about how things matched up, you can kind of see that. Okay, chlorophyll-based mitochondria. The histones we've talked about. Maybe not a big deal to you, but my master's degree was in looking at histone chemistry. And anyone who wants to talk about what's really interesting with histones is that we have the most conserved proteins. Of the proteins in your body are the histones. And your histones have not changed from the time they evolved, like from the pea plant and stuff. You have similar histones. And yet during spermatogenesis, some of you, the sperm kicks out histones and puts in a new set, and different groups of organisms phylogenetically put in different sets. And that was the whole idea. Well, you know, you can track DNA, you can track lipids. What's this deal with histones and spermatogenesis? That was my evolutionary question, so I always like to talk about histones. Okay, muramic acid doesn't seem to be a lot of information there, although we've talked about this in the mycobacterium. Things start a little bit the same here, a little bit different there, and here's the promoter structure, very familiar to many of us. So we see similarities, we kind of see how the tree and the domains give rise in some of the arguments for and against, but even on this very macro level, I've been breaking it down to genes. So I like this. I would, oh yes, and sensitivity to antibiotics. Archaea don't get knocked out by them, and certain ones don't knock out eukaryotes. So I like this because it summarizes so much of the cell biology and the structures that we've talked about up to this point, that you say, well, why are we laying all this groundwork? And it really helps to make sense when we start, as we're standing at the gate, starting to look at this amazing diversity that we're going to do for the next few weeks. So I think it's a really nice summary type of table. I would not spend your days and nights memorizing it, but can you make some sense out of this? Can you see the basis of domains and maybe some of the implications from some of these adaptations and what might have happened to give rise to them? Here, okay, so now we're back to Fort Delisle. So everything I told you, it could be a lie. Okay, why am I paying good money to sit and listen to this bozo? I don't know. You signed up for the class, sorry. This is what I talk about. So Fort Delisle says, okay, if it happened once, there's evidence for a lot of crossing over. What's evidence that we have? Translation. Not translation, transduction. Okay, we talked about genes moving around through viruses, genes moving around on their own. We have jumping genes. Stuff could be really, really mixed up. The more cynical of the microbiologists say, they're all really just one species. There's just variants on that. You don't even know what a species is. How can you even talk about? Yeah, okay, I get reductionistic thinking. Okay, great. On the other hand, it doesn't really help me in my study, whether that study is trying to cure a disease or a study that's trying to figure out what in the world's growing in the Amazon River for whatever reason I may think that's interesting. I kind of think it is. So I like it. It's there. I think Doolittle's right. But I don't know what to do with this information except, wow, that is something to consider. And others have considered. This is one, I don't know how much you can see here, but people are saying, you know, there's other ways to break this down. I was talking with a biologist in Brazil last year, and I was saying, you know, we never really have expressed how the 5 different ways of energy transfer in the prokaryotes really deal with adaptation in ecology. We're fixated with photoautotrophy and chemoheterotrophy, you know, plants and animals. Okay, we don't know. So that's right, you know, and there's evidence other people have thought about that. So he and I talked about that. This essay, now dated, I'm sure it's been updated. I like it because they're saying, you know, there is quite likely a lot of horizontal gene transfer, and a whole bunch of it doesn't work. We know the odds on that. That doesn't really change too much. But so, you know, you can look at kind of symbiosis and maybe secondary symbiosis situations in the protists and come up with a different scheme. You know, something that might have a lot more overlap. They say you can also look at some of these different membrane systems and the way they're expressed. Why? Membrane systems are, you know, where energy and exchange is happening, where diffusion is being controlled. They say, well, look, and you, what are these? These are maybe the protists, and we're dealing with the algae. There was the diatoms, the red algae, the green algae, the cyanophyra, the cyanobacteria. You know, it could be variants on that theme too. And again, you and I are saying, unless we go into studying this for a long time, or maybe you can talk about it in an evolution class with Dr. Bonica. Thinks about the prokaryotes as much. The thing is that there are other ways to cut the pie. There are other ways of coming up with dichotomous keys, with the sense of things. And we can entertain them. Your take-home thing, yeah, I'm putting it out there. There are other things to think about. You know, will this be on the test? Probably not. I mean, what am I going to ask you? You know, could we be wrong? Yes. Okay, 10 points. I mean, you know, what can we do on that? But it is worth thinking about, I think. And as much as I personally, you know, get a little bit frustrated because I want the world to make sense too. You know, I've been trying to, you know, we get a doctorate in philosophy. That philosophy is to develop a scheme of how the world works, and we have to defend that scheme. That's what you do as a doctorate. You have to defend it on essays and what you're doing on different levels. It's the way our education works. I like my scheme to kind of make sense. I put little pictures in my scheme, I move puzzles around, and I try to look at them. I'm a three-dimensional visual thinker. So, you know, I like the simple first Ford Doolittle idea, but I can't help but be fascinated about energy and thermodynamics and how that drives the prokaryotic evolutionary system. And hopefully we have a few weeks to think about that next semester when I leave all of it. So now what's happening, about 1 billion years ago, now we got into symbiosis. Now we got energy for differentiation. We know differentiation shows up ahead of time, and so forth. But now we got this paraphyletic problem of the protists around. Then we can get the rise of metazoans and so forth. And so that starts showing up, little critters that we find in rocks and fossils we can actually look at. It's hard to look at a bacterial fossil, although people do try. And so the diversity of life, where we started out with E.O. Wilson, you know, our best idea of what's going on is happening in this period. We're at our 18 to 20% oxygen. I just put 20 up there as a nice round number, but you know, 18% oxygen. You know, we're in there. We're kind of, as we all know, we're showing up a little blip at the end, and goodness only knows what we're doing to the world, but that's now modern history. Won't worry about it. So some of these things, you know, are happening. The protists coming out about 0.6 billion years ago. We start to see the real big radiation within the domain Euchera, more or less. I know you can argue some of these different lineages. Dinosaurs show up, dinosaurs go, and then we have us and the present self. Yeah, okay. a nice summary statement. I try to put notes in that say things a little bit better than what I say, so that you have something a little bit more tangible than my ideas. So can you look through this and kind of see a summary of what we talked about, purple and green bacteria? Oh, I talked about that. The Earth may have looked real different at one point, even color-wise. You know, periods, I don't get on that too much. I'm not a geologist, so they don't stick in my mind as much. So I deal with kind of the timeline, and that's what I presented. And then, kind of see this. So that's a little summary from your book on a timeline that, as you saw, I created on my own. So, how are we doing? Oh, let's do a little bit of part 4. This is just too much fun, right? Having fun? I can't wait to get out. I know. I'm having fun, so bear with me. At least you are. Is this open? Oh, there it's down here on Chrome. That's right. All right. So where are we at with things? So we want to talk about phylogeny. We have I think we've introduced this several times. It's in your note. We've talked about the idea of the molecular clock. Our big molecular clock in prokaryotic world is the 16 16S RNA. Okay, certain genes, can you do other molecular clocks? Yeah, but we've talked about the 4 basic reasons why we take the small subunit. Review that if you're foggy on it, but it kind of makes sense. It's got the clock has to be within the time span you're looking at. Other types of things, you got to be able to line it up. And so we have it. The sufficient length, yeah. We said, well, we could use 23S, 16S just seemed to work out best, especially early on when we're still running things on gels and stuff. Now we have computers and sequencing. The far surpassed the early Sanger method, and yet the 16S is holding up pretty good in the overall thing. Systematic study of diversity, phylogenetics, the history, taxonomy, how we name stuff. I think at our level we know this. Sometimes it's good to look back on things. So classification, I don't think anything on here is new. I just would like to say 2 great resources from us. I brought up the Burgess manual before. The Burgess manual really came out of clinical studies, and I have the baby's Burgess. Someone stole my baby's Burgess, the pink book I have in the lab. But I borrowed Tracy's, and it's in there. You can look at it. It's mainly, if you look at it, it'll be pages and pages of biochemical tests. Biochemistry is an expression of a gene, so we're looking at the phenotypic type of relationships that were established for culturable bacteria in the Burgess. It's moved beyond that, but that's what it is. Some of you are writing up reports and discussions. Burgess is a great source of information. The other one, has anyone gotten to the prokaryotes? I wrote it out early on. It is like, I'll tell you, you did okay, good. Don't be shy, that's great, good for you. It's a cool book. Each genus has a chapter. Now they're not big chapters, but wow, they tell the ecology, the pathology, the system, okay, more, instead of reading an encyclopedia, you don't want to sit down, most of us don't want to sit down and read the encyclopedia before going to bed at night, but some of us read parts of it. That's how I learned how to make gunpowder, for instance. But the prokaryotes is a great place then to find information on particular species. So it is a reference book. It is huge. And now it's at 4 volumes, and I believe you can get it online. I've ordered it and reordered it for the library. I've lost track of the status. They probably still have print volumes, which are still great for our, we're not leading the edge in speciology here, so still a great resource. I think it was 2 volumes back when I came, now it's 4. Okay, how do we name things? We've talked about that. The International Committee on Systematic Prokaryotes is the group we're trying to get 2 species past, but basically you have to look at the taxonomy, the morphology, the DNA, all these things. I think I've talked about, so I'm going to move on. If anything in here seems I'm making an egregious error by going past it, let me know, but I think that's summary. Now, here is in some place where we get down to definition. Oh, Okay, so we look at the 16S rRNA, I got that, thanks for that. There's 4 rules why I'm doing that, I get that. But you still haven't told me, when do I have a species and when I don't have a species? Okay, can I still look at DNA? Doesn't the RNA, isn't that kind of connected? Yeah, it is. You know, so we come back to that. There is, part 1, we know, okay, too bad, all right. But we gotta do the current definition. Strains, okay, strains are like subspecies, they express differently, but they're not considered to be. Salmonella, many different strains, okay, some will make you deathly sick, some just will make you feel bad, okay. Salmonella's not good, we don't have a good relationship with salmonella, we have to talk to their union. Most important traits include 7% or greater DNA hybridization. Let's stop here. That means if I take species population 1, and it's identical to species 2, let's just say I take E. coli, divide in half, and I took out the chromosome, and I denature it, and then I put the 2 together, I label 1, okay, with phosphorus, and then I put the 2 together, I'll have 100% DNA hybridization, or very close to it, because those were identical members of the same species. So I take an unknown one, and I hybridize it, and, you know, 80% of it's still hybridized. Most of the genes are similar, and I can tell that by labeling. Okay, you know, there's some difference. I don't know what that's all about. Life is big, chromosomes are complicated. I'm gonna let that go. That's still E. coli. Okay, now I take population 1 of E. coli, I label it with phosphorus, or whatever I need to do, P32, and I hybridize it with a culture I got off of the pavement at the Exxon station, because I'm looking for gasoline degraders. And I take that pure culture, and I take the DNA and put it together, and it is only 50% DNA hybridization. That means that only double helix will end up for about 50% of the group at the end. And I'm going to look at the labeled versus the unlabeled, okay. Well, I got a different species there. Now, at first, well, that's pretty simple. I can do that. My sink at home. Yeah, to some extent. You still need something to analyze it. But yeah, that's it. That is the gold standard. Why? Because it goes back to the genome. Why don't we do it more? It's tougher to do. Okay? It's tougher. You got to send it to Germany, and they are not nice about this. I will tell you. But this is what we do more. Now, I'll stop here. I know you're looking at your watch, and I want to keep you late. But this 97% thing is more or less the acceptance now. Some people will say, well, you know, we're more conservative. We're doing 98 or something. But they're saying that if I look at a segment of 16S R DNA, the gene, and it is 97% or more similar to the second thing I'm comparing, that's the same species. Okay. The percent difference beyond 97 tells the different species, and beyond that, the amount of dissimilarity helps to set up where it sits in that phylogenetic tree, because it's a tree of sequence dissimilarity that seems to lead to common ancestors that we can define. And we'll pick it up from there on Thursday. Have a good one. See some